Assalamualaikum, my name is Siti Hajar. Today I want to present about the DNA iteration. So, what is DNA iteration? DNA iteration is routine procedure used to isolate DNA from the nucleus of cells. DNA iteration is a method to purify DNA by using physical and or chemical methods from a sample separating DNA from cell membranes, protein, and other cellular components. The use of DNA isolation technique should lead to efficient iteration with good quantity and quality of DNA, which is pure and is devoid of contaminants such as RNA and protein. Manual methods as well as commercially available kits are used for DNA iteration. Variant tissue including blood body fluids, direct fine neatly aspirated cytology, FNAC aspirated, formalin ferrite, pretreated Ebedu's tissue. Frozen tissue agent can be used for DNA iteration. DNA iteration involves lysing the cell and solubilizing DNA, which is followed by chemical or enzymatic method to remove macromolecules, lipids, RNA, or proteins. So, what can this DNA be used for? Once isolated, DNA can be used for molecular analysis including PCR, electrophoresis, sequencing, fingerprinting, and cloning. So, this is the DNA iteration biotechnical virtual lab. In the experiment, need to include some DNA from a human test subject. So why we need to know the human DNA? Because we to know about the genetic testing, body identification, and analysis of forensic evidence. Scientists include DNA from a variety of reagents. DNA iteration is typically the first step in a layer laboratory process. DNA iteration is important part of the process because the DNA first need to be purified away from protein and other cellular contaminants. We need cell because there where the DNA is. Inside almost every cell in our body is a nucleus, and inside each nucleus is about the 2 meter of DNA. So from the cell, nucleus and chromosome is donated and to DNA. So first we need to collect some cell from out the subject. The skin on the inside of our mouth loses thousands of cells every day. These are the cells we are after. So this is the step we'll follow to purify DNA from a cheap swab as shown below. So this is the material and equipment we will need to purify DNA from cheap cell. Drag the swab over to the inside of the test subject mouth to collect a sample. Next, place the swab into an epidraft tube. Look obviously at the swab. Note there is overhead with 100 of the T-check cell. Inside check cell in nucleus and inside the nucleus DNA. The end of the swab must be cut off so will be able to close the tube. Using the micropipette, add some laser solution to the tube. Laser is a green word to mean to separate. Place the tube into the warm water bath. Alternate the laser solution, just added content to imported ingredients, reagent and enzyme collect proteinate K. The DNA is still wrapped very tightly around protein called heteronase, and the proteinase K cuts apart heteronase to free the DNA. The cell have stayed in the warm water bath long enough for the DNA to be free from the cell, and we have to remove the swab from the tube. Add some concentrate salt solution to the tube. The salt causes protein and other cellular debris to clump together. Place the tube into the centrifuge. Use the micropipette to carefully remove the top liquid and place it into a clean tube. The protein and other cellular debris stay behind. Add some isopropyl alcohol to the tube. Investing the tube several times, mix the isopropyl alcohol into DNA solution. Because DNA is not soluble in isopropyl alcohol, it comes out of solution. Now can see clumped DNA from the ice. Place the tube into the centrifuge and click on the lid to close it and turn it on. The DNA sinks to the bottom of the tube. when the liquid is removed and the DNA is allowed to dry, you can see the risk discovered in the solution of the juice.